So firstly, GBIF is a fair and operational infrastructure. So it's an intergovernmental network and research infrastructure that provides anyone, anywhere, free and open access to data about all types of life on Earth. There's a voluntary collaboration with quite a number of participant nodes and a secretariat that is placed in Copenhagen, Denmark, oversees the whole project. So a bit about the participation. We have 40 voting participants in GBIF, 24 associate countries, 43 other associates, and nearly 2,000 data publishers worldwide. And right here is a map where in green we have the most participation, and blue is where we have moderate participation, and white is where we still need to be improving our presence, I would say, in terms of having a global biodiversity and showing all of this data here within GBIF. So just a little bit about the numbers. So these are actual numbers that we just got yesterday. So there are about 2 billion species occurrence records now in GBIF with 64 country participants, 43 organizational participants, 76,000 data sets, about 2,000 publishers of GBIF data, 24 billion, nearly 24 billion downloads per month, and nearly 8,000 papers now that are using the data from GBIF. So the different biodiversity data types that we can take in. So GBIF, as I've just was previously mentioning, has a fairly strong global presence and as well can take in quite a number of different data types. So we take in, for instance, digitized specimens from museums, quite a bit of what Sharaf discussed already. Observation data from both scientists and from citizen scientists is extremely important. Literature data, so literature data from both the present and the past. Remote sensing data from different organizations, from academics, from industry, and as well environmental data. And the environmental data is a little bit newer. And here, just because this briefly came up, with the environmental data, there is a massive amount of metadata that also goes along with this, and which also must be digitized and made available with the eDNA data so you can actually make sense of it. So this is all done with the common standards. So the Darwin core, and then there's the data publishing and indexing, which makes the data discoverable and usable. So ensuring that it fits the FAIR principles. So the data richness levels that are supported by GBIF. So the data richness can be fairly light or quite heavy, actually. So we have data sets with the metadata available. We can even just take species checklists. There is occurrence only data, and there's also sampling event data. So depending upon the type of data, there's a different level of richness that can be associated to it and can be found on the GBIF website. And as well, there's quite a bit of species occurrence with multimedia evidence. And so right now we have 122 million records with taxonomically identified images. So this includes nearly 1 million audio files and about nearly 4,000 video files. So this includes about 48 million different specimens with the multimedia and 69 million human different observations. As well, as we were just mentioning, for the sources of data are the natural history specimens that we've been discussing. So digitized specimens. So here are two examples that can actually be found on GBIF from the Mycology Herbarium here in Oslo, and also from the Vascular Plant Herbarium at NTNU. Both of these are within Norway. And so these data right now, they can tell you quite a bit about it, but what I can also say at this point is that quite a bit more of the metadata and the technical data that needs to be associated to these sorts of collections probably still needs to be made machine findable and also digitized. As well, observation data from the past. So I was talking about old data as well. So here are two different examples of literature from the 19th century on biodiversity observations that were made that can also be found in GBIF. And as well, extremely important for us, are observations from citizens. So eBird, iNaturalis is international crowdsourcing portals. And here in Norway, where GBIF has quite a presence, then we also have Artsdatabanken, which is a national crowdsourcing portal. And all of these different portals are actually can be directly linked to GBIF for the final publishing of the data. And finally, as I was briefly just mentioning, genomic data and eDNA. And so this is growing very, very rapidly within GBIF and within, I think, the other research consortiums as well. And what I should mention here is that it's fairly recent, is that there has been a crowdsourced book now available on how to publish this sort of data. Right now, it's not terribly well standardized. And for instance, a lot of the metadata that needs to go along with it is not fully standardized yet. And so this is just a cookbook that GBIF has now made available on how to try and standardize and how to publish these sorts of data. And now onto the data coverage from GBIF. So as I've previously mentioned, we have pretty strong data coverage, where in white are the areas that we still need to do better. But for instance, in the US, in Northern Europe, in Australia, and in Southern Africa and South America, we have a strong presence. And so I think a lot of people already know this, but there is a fairly strong taxonomic bias towards birds and against insects and small organisms when you think about their relative biomass versus the data and the literature that's available on them. And this is also very apparent within GBIF data, where we have an occurrence excesses, for instance, towards birds, but we have occurrence shortfalls when we're talking about insects and smaller organisms. So this is something that we actually do want to still try and rectify in the future with GBIF. And then data trends. So the species occurrence records of GBIF over time. So these occurrences are in the millions. And again, they are very high for animals, a bit less for plants, and then others, for instance, insects and small microorganisms, quite a bit less. And then as well, the number of species having occurrence records that are accessible through GBIF over time. So the number of species here is in the thousands. And again, you can see a similar trend. And we would like to note that within animals, it's a lot of species occurrence for birds that we have in GBIF. So birds are very, very well represented within GBIF data. And now a note on this, and this is quite related, especially to Sharaf's talks on museum specimens and their digitization. So natural history museum collections probably have something between 1.2 and 3 billion specimens worldwide. And these are incredible sources for research and provide snapshots through time of our world's biodiversity. There's a lot of challenges working with these data. Oftentimes you don't have the associated metadata that you would like to have with it. And obviously there are oftentimes not very many replicates as you would like to have. But still, even with these challenges, people are working quite hard to work with them. Now, GBIF has about 2.2 billion records overall, which includes about 200 million specimens from museums. So right now, we think that there's about 10% coverage of all of these museum collections worldwide in terms of getting them digitized. So we still have a pretty long ways to go. Now, Scandinavia and Norway also, I will say, is actually doing really, really well within the digitization of museum collections. And so this is just a snapshot of the museum collections at the Natural History Museum where I work. And so we have approximately 6 million specimens total. And so you can see that we've actually done a pretty good job with our digitization, where we have 83% of our botanical collection digitized, only 20% of the zoological, so we need to do better with that. But then again, 63% of our geological collections, which are less interesting for BioDT, but are overall extremely interesting for the natural history museums and understanding world's diversity in general, as well as about 50% of our paleontological collections. So this is a big effort on our part to be pushing this, and this is a continued thing that we're working quite hard on at the museum. Now I was also just talking about some of these challenges. So one way that of digitizing these collections. So GBIF in the past has actually had a crowdsourcing platform, something called Dugnad. So Dugnad is a term within the Scandinavian language that has to do with bringing together people for the societal good. So we've actually had a website where people can where we have these snapshots of metadata, and people have actually helped us with transcription and digitization of all of these labels, which has been massively helpful and actually increasing the speed at which we're getting these things digitized. GBIF data publishing. So it's not that difficult to publish your data with GBIF, but there are some things, of course, that are necessary. So the step one is the digitization of the data, the collections, the herbaria, whatever multimedia file that you already have, which is, of course, digitized, but that is a first step. Step two is the registering for endorsement within GBIF. Step three, and highly critical, is conversion so that it's compatible with the Darwin core format. And step four is publishing from the national GBIF node. Now, alternatives for this can be publishing from the regional GBIF cloud data repository, as well as many citizen science platforms already publish data directly in GBIF. So iNaturalis, as I mentioned, and for instance, here in Norway, Artsdatabanken already work quite closely with GBIF to ensure that they get their work published within it. And now a bit on the data use. So GBIF has a pretty strong policy links with their work. So with the Convention on Biological Diversities, as well within UN sustainability goals. And so here we have one snapshot of how GBIF data in Norway is used within the Convention on Biological Diversity. And so these are some policy goals with the AKE targeted supported biodiversity indicators, where they have actually come back to us telling us how GBIF Norway is doing in supporting their goals in supporting biodiversity. So GBIF Norway is seen as supporting, for instance, trends in invasive alien species through global registration of introduced and invasive species. GBIF Norway can support the Species Protection Index and Protected Area Representative Indices. It supports the comprehensiveness of conservation of socioeconomically and culturally valuable species, including the Agrobiodiversity Index, as well as the Crop Wild Relative Index. And finally, GBIF Norway is seen as supporting the growth in species occurrence records that are made accessible. And that one is not too surprising, but because that is within GBIF's overall mandate is to have these species occurrence data. But that's also very closely tied to these international policies that we're also trying to support. And as well, besides the policy links, GBIF is quite important for supporting research and sustainable development. So for instance, within conservation, protected areas, threatened species, invasive species risk, within food security, crop wild relatives, in situ and ex situ conservation of genetic diversity, fisheries planning, the data is used there. Within climate change, modeling impacts on species ranges, adaptation strategies, mitigation benefits and risks, and as well as in human health. So disease risk based upon occurrence of vectors, hosts, reservoirs, medicinal plants, hazards, and for instance, as snake bites. And so within BioDT, we're helping out with at least 3 of the different user cases related to this conservation within food security and with human health. So there's a lot of data within GBIF that is very important for the BioDT project specifically. And here, just briefly, upon the peer-reviewed publications that are using the GBIF-mediated data. So we're doing quite well with this, upward trends. And so currently, there are more than 3 papers per day that are published using GBIF data. There's been well, we're not quite done with 2022 yet. I'm not exactly certain when this was published, but the trends are going exactly when we were hoping. So GBIF was established in the early 2000s, I think 2003, and we've grown significantly in our presence also within the academic community, as well as our links to the policy that we've already discussed. And just very briefly, because this is actually quite important, is the data citations within GBIF. It's very easy to cite data from GBIF. And this is extremely important actually when we're trying to get people to publish data, especially getting academics to publish data on GBIF. Making it easy to cite their data and for them to get credited is actually extremely important for us. So one can simply download the data from GBIF. They receive a recommended citation with a DOI, and this DOI is cited within the published research or other work. It's really actually hard for me to emphasize enough, especially for the academic community, how important this is to make GBIF data and data that we upload easily citable. And as well, this findable, accessible, interoperable, and reusable, these FAIR principles that we discuss. FAIR data is also about machine readable data. So research and museums having their data on the website in a way that it is reusable typically means machine readable. And so we've already had quite some discussions about this within the BioDT user cases as well, is making certain that the data that we want to use within the user cases, for instance, is fair data and that it is machine readable, because otherwise within BioDT, it's going to be hard to use these data. And this was actually something that came up with within the last user case workshop, where we had a fair number of questions about whether all the data that we plan to be using within BioDT was fair data. And this is something that we do need to try to hopefully manage. And so just very briefly, a new tool within GBIF is something called Bionomia. So Bionomia is looking to better link, I should say, natural history museums to the world's collections. So what we're trying to do here, for instance, is make certain that GBIF data is just simply better integrated with some of these other websites. And as well, and interesting, when you're thinking about a lot of these museum data, oftentimes we don't know the primary authors within the data that's actually published online. You're talking about people from the 19th century oftentimes. And so Bionomia actually has a real case here of trying to ensure that the original scientist that actually collected the specimen can also be found within the metadata that is collected within any sort of specimen that has data within GBIF.